Hey guys, welcome back to my channel. The topic for the video is hemoglobin. So we'll discuss regarding hemoglobin. The first point is what is hemoglobin? Hemoglobin is iron containing coloring matter of red blood cell that helps to carry respiratory gases throughout the body. Hemoglobin is the shortened word that came from hematoglobin, which roughly translates to blood grain, referring to the protein in the blood, which is your hemoglobin. So hemoglobin or hematoglobin are one and the same. Hemoglobin is exclusively found in red blood cells, meaning no other cell in the body can have hemoglobin. So hemoglobin is found inside red blood cell. The number is about 200 to 300 millions of cell per RBC. So this makes up to 95% of dry weight of RBC is hemoglobin. Function of hemoglobin. So hemoglobin carries oxygen in the form of oxyhemoglobin from lungs to the tissue. And on its way back, it will take carbon dioxide from the tissue and it will carry it to the lung for the exchange of the gases. Second function is that hemoglobin acts as a buffer. It maintains acid-base balance of the blood. The third function is hemoglobin gives the red color of the blood. The red color appearance of the blood is due to the presence of hemoglobin in the blood. The next point of discussion is normal content of hemoglobin in the blood or hemoglobin levels. On average, hemoglobin level in the blood is around 14 to 16 gram per deciliter. But this value is varying depending upon the age and sex of an individual. So for infant, the value is about 17 to 22 gram per deciliter. For children, it's around 11 to 13 gram per deciliter. For adult male, it's around 13.5 to 16.5 gram per deciliter. For adult female, the value is in between 12.5 gram per deciliter to 14.5 gram per deciliter. If an individual have reduced content of hemoglobin in the blood, then it is treated as anemia condition. We'll discuss about anemia and the type of anemia in my upcoming videos, so stay tuned and consider subscribing to my channel. The next point is structure of hemoglobin. Hemoglobin is a conjugated protein, meaning it is made from the protein as well as non-protein part. So in the image, you can see this is your globin, which is your protein part of hemoglobin. Whereas this yellow color is the heme, that is your non-protein part. Hemoglobin is a tetrameric protein. Tetrameric meaning having 4 subunits. So here I will draw a simple representation. So we have 4 subunits and in between at the center there is the heme group. So this is your heme and these are your globin chain or the protein part. Each subunit have a heme group and a protein chain. So there are 4 types of protein chain, alpha and beta. Some author write it as hemoglobin is made up of 2 identical dimer, alpha 1 beta 1 dimer and alpha 2 beta 2 dimer. These tetrameric subunits are held together with non-covalent interactions. As hemoglobin is a globular protein, so it will have the hydrophobic interaction at its center. So it will try to hold the hydrophobic interaction at its center. So you can study this interaction into 2 broad categories: within dimer interaction and in between dimer interaction. So in between dimer interaction are mainly hydrogen bond and ionic bond. As at the center of molecule there are hydrophobic interaction, this interaction will form hydrophobic pocket where the heme can get attached and each heme of the subunit can bind with one oxygen molecule. So 4 oxygen molecule per hemoglobin molecule. So now let's study the structure of globin. Globin consists of 4 polypeptide chain of primarily two different structure, as you can see here, alpha and beta. Depending upon the sequence of amino acid and the type of amino acid, there are slight different variation of globin chain. You can see alpha chains, beta chains, gamma chain, epsilon chain, delta chain, and zeta chain. So this is your zeta chain. So in these chains, what are the difference? So there is the difference of amino acid. The number are differ. In alpha, there is 141, whereas beta, gamma and delta 146 amino acid. There might be change in the sequence of amino acid. For alpha, the valine is the first amino acid, whereas beta and delta have valine as first amino acid, but for gamma, it is glycine. So these changes gives rise to different type of globin chain in the hemoglobin molecule. What different it will create if we have hemoglobin molecule with different globin chains? For instance, sickle cell anemia. In sickle cell anemia, the hemoglobin have alpha and beta chain, but in beta chain there is a point mutation that leads to substitution of glutamic acid by valine. So valine comes in the place of glutamic acid. This lead to formation of hemoglobin S or sickle hemoglobin. In this type of hemoglobin, when they are exposed to low oxygen condition, this will form elongated crystal within the erythrocytes. So this is called as sickling, which I will explain in the later part of video. Due to the formation of elongated crystal within the erythrocyte, it damages the cell membrane of erythrocyte and sometimes it gets ruptured that leads to hemolysis, that is ultimately anemia. So the point here is to understand that type of chain in the globin molecule will determine the binding affinity of that chain for oxygen molecule. Now let's study the non-protein part of hemoglobin, that is heme. Heme is iron containing pigment. Red color of the blood is due to the presence of heme in the hemoglobin, or particularly you can say iron. So heme consists of iron and a porphyrin molecule called protoporphyrin 9. This protoporphyrin 9 is made up or is consisting of 4 pyrrole ring. These rings are attached to each other by means of methane bridges. Here in the diagram you can see 4 pyrroles. So these are your pyrrole rings. They have attachment with methyl, vinyl and propionyl group. So these pyrrole rings are attached to one another by help of methane bridges. So here are the methane bridges, and at the center you have iron. So iron plus protoporphyrin 9 is your heme. So heme has 4 bond with nitrogen and 1 bond with the proximal histidine residue of nearby globin molecule. Now we will study the binding side of iron. So iron which is present in the heme is of ferrous form, that is Fe2 plus, and we call this form as ferrous form. This is unstable form of iron. This form of iron is also called as loose form. So for this iron we have to fulfill 6 valency. So see, iron will form one bond with the proximal histidine residue of nearby globin chain. This bond will stabilize the heme position at the center of hemoglobin molecule. The ion is not directly bonded with the distyl histidine residue, as it will get converted into ferric form of ion. So this ferric form of ion will not bind with oxygen. It will affect the binding affinity of oxygen for hemoglobin chain and will directly cause problems with transport of oxygen, leading to abnormalities. Now four bond of iron are with n of each pyrrole ring, that is nitrogen, and one bond is with the oxygen, so that is your oxygen binding side. So one with proximal histidine, four with pyrrole ring, and the one is your oxygen binding side. Now we'll study how oxygen bind with hemoglobin on the binding side. So we will study this by diagram. So here you have subunits of hemoglobin. So this is the first dimer, alpha 1 beta 1 dimer, and at the center you can see in the red color structure is your heme. So this is your hemoglobin molecule. As I mentioned that we have two type of interaction, in between dimer interaction and within dimer interaction. So this is your within dimer interaction. Within dimer interaction is hydrophobic, and with the help of zigzag lines I will show you in between dimer interaction, that is hydrogen and ionic bonds. This form of hemoglobin is called as T form of hemoglobin, or tout, T-A-U-T, tout form, or you can call it as tense form. It is found in low oxygen affinity, and you can call it as deoxyhemoglobin or reduced form of hemoglobin. So when oxygen is available for binding, then it will bind with the heme group, and this oxygen will break some of the hydrogen ionic bond. So it will disturb this bond and it will form this structure. So here we have subunit hydrophobic bond. And you can see some of the hydrogen and ionic bond are disturbed by the binding of oxygen. This form of hemoglobin is your R form, or relaxed form of hemoglobin, also called as oxyhemoglobin. So here you can see there is conformational structural change from T form to R form. Here you can understand one important point, that is cooperative binding or allostric effect. So when oxygen binds with one of the heme group of the subunit of hemoglobin, it will disrupt the interdimer interaction, meaning it will break the hydrogen ionic bond, and it will make easy for the rest of the subunit to get oxygen. So it will increase the affinity of oxygen for the remaining subunits. To understand this effect better, we will use this image so you can understand what is cooperative binding. As it exhibits cooperative binding, hemoglobin is called as allostric protein. So see here we have hemoglobin. The four subunits are shown in the first diagram. One of the heme group from this subunit binds with oxygen. This binding will lead to structural change in the hemoglobin molecule, or you can say conformational changes that widens this gap. So it would be easy for the second oxygen molecule to bind with the second subunit. As this subunit binds, it widens again. So third molecule can easily bind. This will lead to conformational changes. Again, the fourth molecule can easily bind with the fourth subunit. So you can say it is easier for the fourth subunit to get its oxygen compared to that of first. So this type of binding is called as allosteric or cooperative binding. Now let's study type of hemoglobin. There are several different type of hemoglobin found in human. So hemoglobin is heme plus globin. Heme part of the hemoglobin is almost same in these types. The physical and chemical changes in the hemoglobin are due to difference in the composition of polypeptide chains of globin part of hemoglobin. The amino acid sequence of polypeptide chain are determined by the globin genes. Mutation in these genes will lead to structural differences of polypeptide chains. So mutation will cause different varieties of hemoglobin. Most of the mutation does not result in abnormalities. So this type of hemoglobin are regarded as normal hemoglobin where no abnormalities are seen, whereas few of mutation result in abnormal type of hemoglobin. So these abnormal hemoglobin are mutant pathogenic form of hemoglobin. We'll study abnormal hemoglobin as hemoglobinopathies and hemoglobin that are found in thalassemia and related diseases. Type of normal hemoglobin. In human being, there are several type of normal hemoglobin and they are part of the developmental changes from the fetal life to adult. So at the time of birth or during the fetal stage, you can see fetal hemoglobin, which is HbF. Around 80% of the hemoglobin at the time of birth is your fetal hemoglobin. It is structurally different from the adult hemoglobin, as in fetal hemoglobin there are two alpha chain and two gamma chain. Other normal varieties of fetal hemoglobin are Goer 1, which is your alpha 2, epsilon 2, Goer 2, which is zeta 2 chain, and epsilon 2. In Portland, you have Portland 1, which is zeta 2, gamma 2. Also, you have Portland 2, which is zeta 2, beta 2. These hemoglobin variants are very much unstable and they break down in stress. Most unstable fetal hemoglobin is Portland 2. Type of adult hemoglobin. So there is a replacement of fetal hemoglobin by adult hemoglobin, and that starts immediately after birth, and it is completed at about 10 to 12 weeks after birth. So persistence of fetal hemoglobin beyond the age of 4 to 6 months after birth should be raised as suspicion of disordered synthesis of hemoglobin A. That is due to the deficient production of alpha and beta chain. You can call that as alpha thalassemia and beta thalassemia, which we'll discuss in abnormal type of hemoglobin. So for adult hemoglobin, we have hemoglobin A, which is also known as adult hemoglobin, and this is predominantly seen in the blood, and it has two alpha chain and two beta chain. It's around, the composition is 97%. Hemoglobin A2 is also a normal type of hemoglobin that have alpha 2 and delta 2 polypeptide chain. We also have around less than 1% of fetal hemoglobin in adults, and HbA2 is around 3%. So you can see here variation of hemoglobin with age. So at the time of intrauterine life, you can see 94% is your fetal hemoglobin, whereas 6% is your adult hemoglobin. So what I'm trying to say is that HbA, the adult type of hemoglobin, alpha 2 beta 2 chain, appears in the fetus after 20 weeks of intrauterine life, that is 5 months, when the bone marrow begin to function as hemopoietic agent. At that time, HbA appears in the fetus, and after that, at the time of birth, it is around 20%, and rest will be your fetal hemoglobin. So slowly, there is replacement of fetal hemoglobin with adult hemoglobin, and as I said, after 4 months of postnatal life, if you see there is persistence of larger number of fetal hemoglobin, then there is concern that it might be deficient to produce alpha or beta chain, leading to alpha or beta thalassemia. So this was all for normal hemoglobin. Now let's study abnormal hemoglobin. Abnormal hemoglobin. There are two major type of inherited disorder of hemoglobin in human being. First is hemoglobinopathies, and second is hemoglobin in thalassemia and related disorders. So hemoglobinopathy or hemoglobinopathies is a genetical disorder caused by abnormal polypeptide chain of hemoglobin. Whereas thalassemia, in which polypeptide chains are normal in structure but are produced in decreased amount or are absent due to defect in the globin chain. So here there are abnormal polypeptide chains, whereas here synthesis of polypeptide is reduced. So you can call this as qualitative defect, whereas this type is quantitative defect. So first we'll discuss hemoglobinopathies. Hemoglobinopathies are genetical disorder caused by abnormal polypeptide chains of hemoglobin. So first hemoglobinopathies which we are going to discuss is hemoglobin S, abbreviation HbS. So we have two alpha chains and two beta chains. Alpha chains are normal here and the affected chain is your beta chain. So what is the change here? What is the mutation? At sixth position of beta chain, glutamic acid is replaced or is substituted by valine. So due to this mutation, we will have HbS type of hemoglobin. So we have heterozygous HbS and homozygous HbS. So for heterozygous HbS, only one gene of beta chain is affected, while the other is normal. So the erythrocyte of heterozygous will contain both HbA and HbS. So around 40% of hemoglobin would be HbS, rest will be your HbA. And the disease here is referred as sickle cell trait. The individual of sickle cell trait lead a normal life. And they usually do not show clinical symptoms, but at extreme conditions of hypoxia and low pH levels, they will show symptoms. But this individual will live normal life. And this individual will have resistance to malaria. As you know, malaria is a parasitic disease caused by Plasmodium falciparum. Increased lysis of sickle cell will interrupt the parasitic cell. So sickle cell trait can be appear as adaptation for survival of individual in malaria infected regions like Africa. Now homozygous HbS. Here both the gene that code for beta chain are affected. So we will have HbS type of hemoglobin. And this HbS type of hemoglobin is found in sickle cell anemia disease. And this individual unfortunately cannot live beyond 20 years. So the life cycle is less than 20 years. So we'll discuss abnormalities associated with HbS. So we have HbS hemoglobin due to mutation in the HbB gene. The HbB gene is present on chromosome 11. So there is a point mutation on this HbB gene. So at sixth position on the beta chain, the glutamic acid is replaced by valine. Glutamic acid is hydrophilic in nature, whereas valine is a hydrophobic amino acid. So due to this change in nature, it will decrease the solubility of HbS in deoxygenated form. See, we have hemoglobin in two form. When it binds to oxygen, it is called as oxy-HbS. And when oxygen is not there, it is reduced hemoglobin or deoxyhemoglobin. So here we have oxy-HbS and deoxy-HbS. So due to this change in nature, there will be decrease in solubility of deoxy-HbS, whereas solubility is not affected for oxy-HbS. So why it is so? See, the substitution of valine for glutamic acid result in a sticky patch on the outer surface of beta chain. So let's see by diagram. Suppose this is your oxy-HbS. It will have 2 beta chain and 2 alpha chain. These are our 4 subunits. These sticky patch are present on the outer surface of beta chain. So here are your sticky patch. This is your deoxy-HbS. Same, it will have 4 subunit, beta, alpha. The sticky patches are also present on the deoxy-HbS on the beta chain surface. So see, here we have the sticky patches. These are your sticky patches. There is a site or receptor complementary to this sticky patches. It is present on deoxy-HbS. So these are your receptors. And this receptor are masked in case of oxy-HbS. Although sticky patches are present on oxy-HbS, molecules of oxy-HbS cannot bind among themselves or with the molecule of deoxy-HbS. Whereas this deoxy-HbS will form long strands or what we call fibers of deoxyhemoglobin, or it is also called as sickling. So let's study mechanism of sickling.
Under condition of
Under conditions like hypoxia, acidosis, and dehydration, deoxy-HbS, see, the sticky patch of one deoxy-HbS will bind with the receptor of another deoxy-HbS, and this process result in formation of long aggregate molecules of DHbS. See, this is one deoxy-HbS plus one more deoxy-HbS. So see, so more and more deoxy-HbS will bind to one another, forming long fibers precipitate. So this is called as polymerization of deoxy-HbS. This stiff fiber distort the shape of red blood cell into sickle shape. And this sickle shape erythrocytes are vulnerable to lysis. The cell membrane is very fragile and it ruptures, so there is continuous breakdown leading to lifelong anemia. And this sickle cell, when passing from capillaries, will block the capillaries, resulting in poor blood supply to the peripheral tissue, leading to excessive damage and inflammation of certain tissue, causing pain. And there will be increased susceptibility to infection, and individual with sickle cell anemia die before they reach adulthood. So the lifespan is very small, it's less than 20 years. As during this condition of hypoxia, acidosis, and dehydration, there is sickling, that is due to polymerization of deoxy-HbS. So if this HbS is maintained in oxygenated form, the sickling can be prevented. So management of sickle cell anemia include administration of sodium cyanide, inhibits sickling in the erythrocyte, as cyanide increase oxygen affinity to HbS, lowering
the formation of deoxyHbase. However, there are certain side effects like peripheral nerve damage. So, in patient with severe anemia, blood transfusion is required, but this blood transfusion increases the risk of iron overload and can lead to sclerosis of liver. Replacement of HbS hemoglobin with other form of hemoglobin can reduce sickling. So, fetal hemoglobin reduces sickling, and sickle cell disease awaits gene replacement therapy. Moving on, the next hemoglobinopathy is hemoglobin C, abbreviation HbC. So here, alpha chains are normal, the beta chains are affected. At the sixth position of the beta chain, glutamic acid is replaced by lysine. HbC hemoglobin is found in people with hemoglobin C disease. It is also called as Cooley's hemoglobinemia. Both homozygous and heterozygous individual of HbC disease are known. This disease is characterized by mild hemolytic anemia. Moving on, we have hemoglobin E. In hemoglobin E, the beta chains are affected. So at the 26th position of beta chain, glutamic acid is replaced by lysine. Hemoglobin E disease is the most common after HbS. The individual of HbE, either homozygous or heterozygous, have no clinical manifestation. This variant is very prevalent in West Bengal in India. The last is rare form of hemoglobinopathy, which is hemoglobin M disease. Hemoglobin M disease is characterized by presence of hemoglobin M. This altered form of hemoglobin is due to point mutation that occurs in the globin encoding genes, mostly involving tyrosine substitution for proximal or distal residue of globin chains. As a result, the heme get oxidized in hemine, where the ion of heme, which is in the ferrous form, get oxidized into ferric form Fe3 plus. As I mentioned before, the ferric form does not bind with oxygen, so the binding affinity of oxygen is decreased, and this would lead to cyanosis. Hemoglobin M disease is rare disease, and it's not that life threatening, and no effective treatment is found. So there are several other altered form of hemoglobin like hemoglobin I, hemoglobin J, hemoglobin D. So here I would like to mention hemoglobin D Punjab. Hemoglobin D Punjab is the commonest hemoglobin variant seen in the Punjabi population, and here there is replacement of glutamic acid by glutamine at 121 position of beta chain, as you can see here in the table. So in this table you can recall important hemoglobinopathies. One more important point to remember is about HbSD disease. This is a severe condition. People with this disease have red blood cells that contain both cecal hemoglobin, that is hemoglobin S, and hemoglobin D. The type of hemoglobin D usually determines the severity of this disease. All of these abnormal hemoglobins can cause hemolytic anemia and splenomegaly, that is enlargement of spleen. So this was all for this video. In the upcoming video, we'll study derivative of hemoglobin. Thank you for watching.